Welcome to week 6. This week we're going to cover chapters 11 and 12. In chapter 11 we'll focus on DNA replication, recombination, and in chapter 12 we'll talk about DNA organization into chromosomes. So to begin with, when we talk about DNA replication, we're talking about the process of copying a DNA molecule. This replication is known to be semi-conservative. And what we mean by this is that each strand of the original double helix is the parental molecule serves as a template for the new strand in a daughter molecule. So let's look at that a little closer. Here you can see the parental DNA double helix, nicely paired up. And then you can see where this is split into two strands. The pink is the parental strand, or old strand. The new strand here is indicated in blue. So what you can see is at the end of replication, you're going to have two strands of DNA instead of one strand of DNA, and each of these is going to have one of the old parental backbone strands and one of the new daughter strands that's created. So there's three possibilities. When they realized how DNA was being replicated, they thought about DNA replication as being conservative. In this case, what would have happened is that the original helix would be conserved and two newly synthesized strands would come together. Semi-conservative, which is what we now know is how it happens, in this case each replicated DNA molecule consists of one old strand and one new strand. And then lastly, dispersive was another possibility, in which case the parental strands are dispersed into two new double helices. And let's talk about how they discovered this. Okay, well first let's visualize what we're talking about here. If something was conservative, after one round of replication you would have one strand that was completely new and one strand that was completely old. Okay? In semi-conservative, what you can see here is one strand of old, one strand of new. In dispersive replication, the parental strands are dispersed into two new double helices following replication. And in this, each strand consists of both old and new DNA. This mode, however, would require cleavage of the parental strands during replication. It is the most complex of these three possibilities, and therefore it was considered to be the least likely to occur. However, until they actually did experiments with it, they were not able to rule it out as an experimental model. So Michelson and Stahl did most of the experiments showing that DNA replication was semi-conservative. And what they did was they radiolabeled the nitrogen within DNA with a different isotope, 15N. And so if you spun this in a centrifuge, what you would find is one band of very heavy DNA. Both strands would be labeled with this nitrogen. If this replication was semi-conservative, the new nitrogen that they added in would be normal nitrogen, not radiolabeled nitrogen. So what would happen after one generation, if it was indeed semi-conservative, is that you would have one parental strand and one new strand. And after one generation, 100% of the DNA should be this intermediate weight if you applied a centrifugal force to it. And after subsequent generations, again performing the same methods using unlabeled nitrogen, what you would expect is that half of the strands would be completely new or light with this nitrogen, and then half of them would still be this moderate or intermediate weight DNA. And you would not expect to find any that was this heavy DNA. And that's indeed what they saw. And after subsequent generations, many different generations, most of the bands shifted to this lightweight, and there were just very few, there's the two original strands that consisted of that heavyweight DNA. So they also performed experiments showing that eukaryotic DNA replication is also semi-conservative. And this was performed by Taylor Woods Hughes in 1957, and they used broadbean to demonstrate that this DNA replication is semi-conservative. So what they did was they looked at chromosomes, and they first took these unlabeled chromosomes, they labeled them with tritium, this is this H3 thymidine. So all of the hydrogens within thymidine would be labeled with this radioactive isotope. And after this replication, after this labeling, what you would expect to see is the two sister chromatids, both of which would be labeled. And you can see here a diagram of what they might have seen under the microscope. Okay, so after metaphase, what you would expect is that the sister chromatids would be pulled apart during anaphase. Okay, and then at the end, what you're going to see after a second replication is that only one of the chromatids would be labeled. And that's exactly pretty much what they see here. There is, however, some sister chromatid exchange, and so you may see part from one chromosome, part from another, and you can literally see that this homologous recombination happened right at this point right here. In this case here, there was no sister chromatid exchange, no homologous chromosomes synapsing, and you did not have any crossing over. So when DNA replication begins, there is an origin of replication for this. Where this replication is occurring, the strands of the helix are unwound, and this creates what we call a replication fork. This replication occurs in both directions. It is bidirectional, and therefore there are two replication forks. And what we're going to talk about specifically here at the beginning is we're going to talk about prokaryotic DNA replication. So if you look at a typical prokaryotic genome, you can see here again it's circular double-stranded DNA. There is going to be what we call an ORI, in this case an ORI-C. This is the origin of replication. And this is where all of the replication machinery starts. Okay, after replication begins, it happens bidirectionally. So it happens this way as well as this way at the same time. And after time, you can see it unzipping even more. And at the end of this, it's going to reach this termination region, and you will have two complete genomes.